The research began with the discovery of viruses, which do not evoke an inflammatory response, the hallmark of infection. These viruses were referred to as being stealth-adapted. The immune evasion mechanism was explained as the loss or mutation of the relatively few virus components that are normally targeted by cellular immune system. Stealth adaptation is a generic process that can potentially occur with all viruses, including monkey cytomegaloviruses initially transmitted to humans through polio vaccines. Recovery from the cytopathic effect CPE of stealth-adapted viruses was observed in virus cultures. Moreover, clinical recovery could occur in infected patients and was clearly observed in virus-inoculated animals. The recovery was associated with the production of energy-transducing materials, which commonly self-assemble into particles and fibers. Marked disruption of the mitochondria was seen in the surviving cells, both in cultures and in human brain biopsies. This led to the notion of an alternative cellular energy ACE pathway, distinct from mitochondria-mediated oxidative phosphorylation. The energy-transducing materials formed in the recovering virus cultures are also present in dried perspiration and other body fluids of stealth-adapted virus-infected patients. They may directly fluoresce under UV light, and especially do so when mixed with certain dyes, including neutral red dye. I refer to these fluorescing and commonly electrostatic materials as ACE pigments. They are particularly notable in patients diagnosed as having delusional parasitosis or Morgellons disease. They also explain the UV fluorescence seen in herpes simplex virus HSV skin lesions being treated with neutral red dye. The ACE pathway has been equated with activation of the body's fluids and with the long history of various methods of activating water. The physics of water activation is currently ascribed to an environmental energy force called keliokinetic energy, limiting electrostatic attraction. The force is attracted to dipolar materials with separated electrical charges. Many of these materials can then transfer kelia to nearby water. Once water is sufficiently activated, its separated charges can directly absorb kelia from the environment, resulting in further activation. This principle explains homeopathy. Proof of the enhanced biological activity of kelia-activated water has been obtained in agricultural studies. In addition to increased crop yields, studies in both rice and sugarcane show less disease and infestation in the treated fields. The stage is now set to test kelia-activated water in rigorous clinical trials. It should be possible to extend the positive findings of homeopathy by showing similar benefits from consuming kelia-activated water, also referred to as ACE water. Illnesses that should be addressed include those caused by stealth-adapted viruses, including autism, chronic fatigue syndrome CFS, and various mental illnesses (ME). The studies can also include a wide range of illnesses due to an insufficiency of cellular energy ICE from oxygen-dependent food metabolism, such as COP, cardiovascular diseases and metabolic diseases. The increased energy demands of conventional infections, such as herpes, hepatitis, HIV, tuberculosis, childhood diarrhea, and many others, can also be addressed by enhancing the ACE pathway.